Hey everybody, we are going to move on with our metabolism unit, and we're going to start talking about cellular respiration and how we harvest chemical energy. So is this the process of turning food into energy? Yes and no. So this moose isn't going to be able to extract energy directly from the grass, but once that grass has been digested and his body's absorbed the nutrients, we're looking at what happens when the glucose and the carbohydrates that this moose has absorbed, how they get broken down and how energy is released from that process. So here is our overall equation for cellular respiration. We have C6H12O6 and 6 oxygen, so glucose and oxygen. And at the end, our products are going to be 6 carbon dioxide, 6 water, and energy. So the energy is released from the chemical bonds in those complex organic molecules. So as that glucose is broken down, energy is released, and that's energy that the cell can use to do work. So just kind of a preview of the things we're going to talk about with respiration. We'll focus on how energy is released from food. We'll talk about how food stores energy in chemical bonds, and then we're also going to talk about ATP again and how this is usable energy for cell work. So the focus of this chapter, we're going to look at what is this reaction supposed to do, where does this reaction and the parts of this reaction take place, what is needed for each stage of cellular respiration, and also what is produced at each stage of cellular respiration. Before we get into the details, though, we want to talk about oxidation and reduction. So oxidation means that you're losing electrons, you're losing energy, or you're losing hydrogens from carbons. And so when we talk about cellular respiration, we can look at things like oxidation and reduction and how these two are usually paired together. Reduction then is when you gain electrons, gain energy, or you gain hydrogens to carbons. So be careful with this, because typically when you think of reduction, like a reduction in price, you're usually excited because the price has gone down. Here though, we're talking about gaining of electrons, gaining of energy, or gaining of hydrogens to carbons. So just understand that it is different than how you use reduction in sort of lay everyday terms. So here's an example of a redox reaction. And so you've got your methane right here, there's C4, and then you've got 2 oxygen. All right, those are the reactants. The products of this reaction are carbon dioxide and water. So the methane is being oxidized because we are taking off those hydrogens that were attached to it. So here we have a carbon with hydrogens attached. Here we have a carbon with no hydrogen, so it's been oxidized. And then the reduction, we have the reduction of oxygen. So here we have oxygen with no hydrogens. Here now we have oxygen with hydrogens attached, so it has been reduced. So this is oxidation and reduction. Again, these reactions are usually linked or paired together. And look for these kinds of things as we study photosynthesis. And a lot of this is going to happen when we talk about phosphorylation. And so phosphorylation, we mentioned it in chapter 8, is anytime you add a phosphate group to a molecule. And when that phosphate group is added, it's going to add energy to the molecule for chemical reactions. What we're looking at here is an example of one type of phosphorylation. So this is substrate level phosphorylation. And so what that means is you have a substrate here that has this extra phosphate group. Okay, so this substrate is going to attached to an enzyme, and here you also have ADP, so you have an adenosine with 2 phosphate groups. Now while these two are attached to the enzyme in the active site, what's going to happen is this ADP is going to become phosphorylated to ATP, and so afterwards when the enzyme releases all of this, you now have your product minus the phosphate group, and now you have a third phosphate here making ATP. So this is an example of substrate level phosphorylation, which is one of the types that we're going to study in this chapter. Okay, so now we've got the background out of the way. We are going to look at the 3 parts of cellular respiration. So the first reaction that takes place is called glycolysis. After that we have Krebs cycle, and then finally the electron transport chain. So we're going to look at each of these 3 parts individually. Today we're going to focus on glycolysis. And so again, you've got this glyco part of the word up here. It looks like glucose. And then the lyse, meaning to split. So glycolysis literally means to split glucose. This is a really important step. It's a universal step in all types of respiration. And it's thought to be a really early type of cell energy process. And the reason for that is because it is universal. So it's used by all types of cells. The function of glycolysis, again, is to split glucose and produce NADH and ATP. Now, ATP we have talked about. NADH we'll get to in a second. And the location of this reaction is actually in the cytoplasm. So when we talk about cellular respiration, we'll talk a lot about the mitochondria, which is where Krebs cycle and electron transport take place. But understand that glycolysis happens outside of the mitochondria in the cytoplasm of the cell. And so here we have our glycolysis. So the yellow area is the cytoplasm. The whole point of glycolysis is to break glucose down into pyruvate. You're actually going to get 2 pyruvates for every 1 glucose because glucose has 6 carbons and pyruvate has 3 carbons each. So you're literally just breaking it in half. In terms of the number of carbons. And then ATP is produced here as a part of this reaction, and it's produced through that substrate level phosphorylation that we just talked about. Now, the rest of the steps of cellular respiration that we're going to talk about later, they all take place in here inside the mitochondria. All right, so I talked about this NAD plus and NADH, and we're going to kind of explain that now. So NAD plus is an electron carrier compound, and the whole point of this molecule and other molecules like it is to shuttle electrons around within the cell. Now, I talked about NAD plus and NADH, and so these electron carriers exist in 2 forms, an oxidized form and a reduced form. So what we have here is the NAD, and the NAD plus is going to be your oxidized form. When you add the electrons to it, then it becomes the reduced form. And so NAD plus and NADH are going to go back and forth. So when this NAD plus picks up 2 electrons, it becomes NADH. When the NADH drops those electrons off in a different part of the cell, it will go back to being NAD plus and it can pick up more electrons again. Okay, so back to glycolysis. What exactly do we need for glycolysis? Well, we obviously need glucose because we need to split it in half. And just to get glucose started, we're going to need 2 molecules of ATP. Then we also need 4 molecules of ADP so that they can become energized. And then we're also going to need 2 NAD pluses to carry electrons to a different part of the cell after glycolysis. Now, after all of this, what is produced? Well, the 1 molecule of glucose will get split into 2 pyruvic acids or pyruvates. The 2 ATP will become ADP because they will have donated their energy. The 4 ADPs will have picked up a phosphate group and they will become ATPs. And then the 2 NAD pluses will have picked up electrons and they will have become 2 NADH. So now the net result at the very end of all of this, we are going to have a surplus of 2 ATP for every glucose molecule and 2 NADHs that now have electrons that they can carry to another part of the cell. So here is something that we talked about in class before, this idea that you've got to spend energy to make energy, kind of like the whole you have to spend money to make money. So in order to actually get energy, and go through this process of glycolysis, you need to spend some energy first. So here we start with glucose, and we're ending with 2 pyruvates, okay? But in order to get all the way down here, first you need to put in or spend some energy. So you need to put in 2 ATPs. These ATPs will then become ADPs because they're going to phosphorylate something, provide some energy, and then over here they will be without those phosphates. Now, once this reaction gets underway, you are also going to produce 4 ATP. Okay, so this is where you're making some energy. And you will also create 2 NADHs from NAD+. So this molecule is going to grab onto 2 electrons and hydrogen, and it's going to make some NADH. Now, you're spending 2 ATP initially up here, so you're spending this energy, but then you're making this energy. So if you're spending 2, but you're gaining 4, 4 plus negative 2 is going to be 2. So you have a total or net gain of 2 ATP. So that is it for glycolysis right now. I want you guys to take a look at this lecture question and shoot me an answer from the website. So why is glycolysis considered universal? And why does it take place in all cell types, even bacteria that don't have organelles? All right, guys, thanks.